Cher docteur, chères mesdames et messieurs, nous vous remercions d'avoir accepté notre invitation à ce webinaire interactif sur la photobiomodulation pour le traitement de la DMLA atrophique. Je m'appelle Attila Kaplanzi et je suis le directeur de vente de Lumitera en Europe. Avec ce webinaire, nous souhaitons vous offrir une plateforme scientifique pour l'échange des expériences et des connaissances que vous pourrez éventuellement utiliser ultérieurement au profit de vos patients. Nous sommes donc ravis d'accueillir deux orateurs, Dr Streho et Dr Clark Tedford. Dr Streho est associé et co-gérant du centre Explore Vision et il a une grande expérience de la photobiomodulation utilisant la plateforme Valeda Light Delivery System. Il partagera cela avec nous dans la seconde partie. Nous sommes également ravis d'avoir ici le Dr Clark Tedford, qui est le PDG et cofondateur de Lumitera. Et dans la première partie, Dr Clark nous informe en anglais du contexte scientifique de la photobiomodulation et de l'état le plus récent des études cliniques en cours et prévues. Dr Clark Tedford est titulaire d'un diplôme en neuropharmacologie de l'Université de Iowa aux États-Unis. Ce webinaire devrait être le plus interactif que possible. Si vous avez des questions ou des commentaires, veuillez écrire-les dans la fonction questions et réponses qui se trouve en bas de votre menu Zoom. Et maintenant, je demande à Dr. Clark pour commencer par la première partie. Clark, would you please start with the first part, please?
Thank you, Attila. And Matea, it's a pleasure to be on the Zoom webinar again with you. We're very excited to talk today about photobiomodulation. I will provide a little background on the mechanisms and the importance of our multi-wavelength approach. Most importantly, I'm going to turn it over to Matea to talk about the top-line data from our US pivotal clinical trial called the LightSight 3. And then we'll open it up for discussion. This is an interactive program, as Attila indicated. We'd love to have questions. You can put them into the Q&A, and at the discussion point, we will try to answer all of your questions. So just as a background, Lumitera is based in the US, in the state of Washington, near the Seattle, across the Sound. And I was formed in 2013 by myself, 2 eye doctors and 2 engineers, with the idea to use photobiomodulation to create a non-invasive treatment to improve visual function, to stop or slow progression of debilitating eye diseases, and hopefully prevent vision loss and blindness. Photobiomodulation, which is the underlying technology we're going to talk about, is also known as low-level light therapy. It's really an expanding technology where a lot of research is being done. It's using low levels of light, typically in the visible to near infrared, to stimulate cellular targets, and thus leading to beneficial clinical downstream effects. PBM is approved and used in a wide variety of areas. It's used in physiotherapy and skeletal muscle for inflammation, chronic pain. It's a tier 1 product in arthritis. Excellent work is going on in diabetics with open wounds to show that it can stimulate recovery processes and close wounds. Athletes are using it in sports medicine to recovery from injury, if your ankle or knees, if you've injured them. And what we're seeing is about 20 to 30 publications per month coming out on photobiomodulation in various areas. The instruments obviously are very specific for the physician in that particular area. And what we have created is an eye-safe instrument called the Valeda. The mechanisms, we have a very nice animation. I'm not going to play it today, but it's on our website. But in essence, we're talking about a mitochondrial-based approach. We're going after an enzyme called cytochrome C oxidase, which is shown in the cartoon on the left. And this enzyme is a large proton pump that's involved in energy production as part of the electron transport system. The proton pump is used to push protons across the intermitochondrial membrane to establish then the gradient that's used for ATP generation by the next enzyme, ATP synthase. It requires oxygen and electrons as the two substrates that bind to one of the active sites that is accepted photons from 660 or far red. The other copper will accept electrons and shuttle them into the enzyme, and that is also amenable to near-infrared light absorption. And so we're really using these two wavelengths to drive and restore mitochondrial membrane potential and to reset metabolic activity. We have a third wavelength, 590, which is far yellow in the visible range, and it's been shown in retinal epithelial cells to inhibit VEGF expression naturally, and it can also improve phagocytosis. So we're using that as a supplement to the other two wavelengths to create a very specific multiple wavelength approach to treating degenerative eye disease. And particularly, our focus has been on the dry form of age-related macular degeneration. Here is the Valeda system, and it sits on an adjustable table, and as you can see, it can be used to treat the patient very comfortably in a flexible schedule that we use. It has approved about 2.5 years now in Europe, and we have been now working on additional clinical trials. In the last year, in 2021, we had 3 trials, including our post-market European multi-center LightSight 2 study completed, initial study in diabetic macular edema and retinopathy patients that was presented at ARVO a year ago, and a study using electroretinogram as a biomarker to show improved retinal ganglion function. What we're going to talk about today is the LightSight 3 US pivotal study, and Matea is going to speak on that. This study will now be presented and discussed with the FDA, and hopefully will lead to US approval at the end of the year or in early 2023. In addition to this, we have 2 other studies that are teeing off. We have a European registry study to collect additional patients for reimbursement in Europe and to support the safety and efficacy of the design trials so far. And we have a NIH study looking at macular edema and retinopathy in the US that is getting ready to tee up as well. So a lot of work is being done by Lumathera and our colleagues out in the retinal community in terms of its mechanisms and its benefits in various diseases. With that, I'd like to turn it over to Dr. Dr. Streho and have him present the LightSight 3 top-line efficacy and safety data at month 13. Matea?
Thank you very much, Clark. It's a pleasure to be with you. Bonjour à tout le monde et c'est un vrai plaisir de pouvoir parler aujourd'hui de photobiomodulation, de Valeda et donc de pouvoir parler de résultats scientifiques. Et tout d'abord, je voudrais rendre hommage à la démarche de l'Umitera. Et depuis le début de l'aventure, ils nous fournissent des résultats scientifiques de grande qualité. Ils partagent avec nous à travers ces réunions toutes les études et ils nous apportent en fait toutes les connaissances à jour sur les dernières connaissances parce qu'il s'agit de nouveautés, il s'agit de choses qu'on apprend au fur et à mesure et donc c'est toujours intéressant d'avoir des échanges de qualité en toute confiance pour pouvoir développer des nouvelles thérapeutiques, donc la photobiomodulation. Alors aujourd'hui, on a la chance, et c'est un honneur pour moi de pouvoir présenter en avant-première en France les résultats de LightSight 3. Donc c'est l'étude LightSight 3 qui fait suite à l'étude LightSight 1, 2 et maintenant 3. Et pour ceux qui suivent l'aventure depuis l'étude LightSight 1, c'est à ce moment-là que j'ai pris le train en marche. On est en octobre 2019 et donc les premiers résultats ont permis de montrer de manière scientifique dans une publication dans Retina l'efficacité de la photobiomodulation. L'aventure a continué et donc aujourd'hui maintenant on a les résultats 1 an, 13 mois de l'étude LightSight 3, qui est l'étude pilote pour la validation de la photobiomodulation pour le marché américain. Donc vous avez ici pour l'étude LightSight 3 les sites qui ont inclus les patients. Il s'agit de 100 148 yeux pour 100 patients. Et donc, je voulais insister sur les différents sites qui ont inclus les patients aux États-Unis. Et vous voyez qu'il y a les très grands noms de l'ophtalmologie américaine, Dianado, Richard Rosen, vous voyez, etc. C'est vraiment une gage de qualité et il s'agit de centres de référence. Next. Alors, ce qui m'a intéressé également et que l'UMITER a continué dans les études, c'est cette approche scientifique rigoureuse. Et donc, on va regarder le protocole d'évaluation de la photobiomodulation dans l'étude Lightside 3, comme on a appris à regarder les médicaments dans la dégénérescence maculaire liée à l'âge. Et donc ici, on a une étude qui est randomisée, qui est masqué, qui est multicentrique, qui est versus contrôle. Et donc, ça donne un niveau de puissance scientifique qui est la plus importante de toutes les études. Et donc, en fait, on a une qualité scientifique irréprochable. Un petit mot sur le principe du placebo. Ici, on a un appareil qui permet de simuler un traitement. Le traitement a eu lieu, mais avec un dosage 100 fois inférieur. Donc, gardez à l'esprit qu'il y a encore un petit traitement, mais avec 100 fois moins d'effet. Comme ça, le patient ne sait pas lequel il a, mais nous, on le sait. Le protocole est très intéressant parce qu'au moment de l'inclusion, on a réalisé donc 9 séances sur 3 à 4 semaines. Donc, en moyenne, c'est ce qu'on fait nous au Centre Expert Vision, c'est le patient vient lundi, mercredi, vendredi, pendant 3 semaines. Et donc, il a ces 9 séances et on va répéter ça tous les 4 mois. Donc, il a une première session de 9 séances sur 3-4 semaines, 4 mois après la deuxième, et ainsi de suite jusqu'à la fin de l'année, au mois numéro 13, où on évalue. Et le critère retenu était le gain d'acuité visuelle par rapport au groupe contrôle. Next. Alors, les critères d'inclusion, donc le critère principal, c'était le gain d'acuité visuelle par rapport à l'inclusion, groupe traité par photobiomodulation versus groupe témoin, et vous allez voir que l'étude a atteint ses objectifs, et de manière assez impressionnante, statistiquement significative, les patients traités ont un gain d'acuité visuelle supérieur au groupe témoin, et donc on va dérouler ça dans la suite. Il y a également des critères secondaires, et c'est des critères secondaires qu'on a appris à connaître maintenant depuis les études Lightside 1 et 2. C'est en général les tests fonctionnels, comme les sensibilités au contraste, l'acuité visuelle en faible luminance, ou des critères anatomiques comme le volume des druselles, la vitesse en enseignement. Next. Alors, au niveau des critères d'inclusion, donc il s'agit donc de 100 patients avec 148 inclus. On a une population qui est extrapolable à notre population traitée en France, 99% de caucasiens avec une prédominance de femmes, l'âge moyen de 75 ans. Et donc, je vous rappelle, on s'adresse à la DMLA intermédiaire. Donc, il s'agit essentiellement de drusènes séreux confluents avec des migrations pigmentaires, atrophie tolérée, mais sans atteinte centrale. Il ne s'agit pas principalement de traiter des DMLA atrophiques au stade terminal. Et ce qui est intéressant, c'est de voir que, comme en France, aux États-Unis, une grande partie des patients qui sont déjà dans la DMLA intermédiaire sont traités par des compléments alimentaires, des vitamines AREDS, ce qui est notre pratique aujourd'hui. Next. Alors, rentrons tout de suite dans le résultat, et donc ça, c'est le critère principal, et le succès de l'étude Lightside 3 a été de montrer, donc vous verrez pour les différents graphiques, en jaune, le groupe témoin, et le groupe traité par photobiomodulation Valeda et le diagramme en vert. Et donc, ce que vous voyez au niveau du gain d'acuité visuelle, c'est que vous avez à chaque étape un gain supérieur, et quand on regarde au 13e mois, donc c'était le critère principal, on a un gain qui est statistiquement significatif, supérieur pour le groupe traité par photobiomodulation. On va encore plus loin, on voit que sur ce groupe traité qui a un gain d'acuité visuelle supérieur, on a en moyenne 5,4 lettres avec un petit p qui est très significatif. Et ce qui est intéressant, c'est que ce gain d'acuité visuelle, c'est ce qui nous a été promis depuis l'étude Lightsight 1, qu'on a retrouvé dans l'étude Lightsight 2, également dans l'étude Lightsight 3. Donc en fait, on sait aujourd'hui que la photobiomodulation apporte en moyenne entre 5 et 6 lettres de gain d'acuité visuelle. Next. Quand on regarde la distribution de ces acuités visuelles, il est toujours intéressant de s'intéresser donc de regarder les patients qui ont des gains d'acuité visuelle et en fonction du gain, vous savez que 5 lettres équivaut à une ligne d'acuité visuelle. Donc on va s'adresser à des patients, 5 lettres c'est déjà une ligne d'acuité visuelle, mais 10 c'est 2 lignes et au-delà c'est 3 lignes, donc c'est vraiment des gains d'acuité visuelle très importants. Et donc on voit que pour la partie des pertes d'acuité visuelle, que ce soit faible ou plus importante, le groupe témoin est supérieur. Et pour ce qui est des gains d'acuité visuelle, on voit que plus on augmente dans les gains d'acuité visuelle, et plus le groupe de photobiomodulation prédomine. Donc on voit l'effet de ce traitement sur le gain d'acuité visuelle. Alors, il est intéressant de voir que ce gain d'acuité visuelle est probablement un critère qui nous parle beaucoup parce qu'on est habitué dans la DMLA, mais si on traite les patients très tôt, le gain d'acuité visuelle ne sera pas énorme. Donc, il ne faut pas forcément chercher à avoir des gains d'acuité visuelle, mais des maintiens. Néanmoins, il est intéressant de voir avec ces résultats qu'il y a vraiment un effet bénéfique du traitement montré sur ces 13 mois. Next. Alors, au niveau de la distribution de ce gain d'acuité visuelle, on voit que 55% des patients traités par photobiomodulation ont des gains supérieurs ou égaux à 5 lettres, comparé à 40,8 pour le groupe placebo. Alors, on peut se demander pourquoi le groupe placebo a des gains d'acuité visuelle. C'est là où c'est important de se rappeler que la photobiomodulation dans le groupe témoin a quand même un traitement, mais avec une puissance 100 fois inférieure. On a 26,4% des patients qui ont un gain supérieur à 10 lettres, donc 2 lignes, ce qui est déjà un gain très impressionnant, et on a 5,5% des patients qui ont des gains supérieurs à 15 lettres. Donc ça, c'est vraiment des choses marquantes, perçues par les patients, très très bien perçues par le praticien, et donc qui apportent des vrais gains fonctionnels pour le patient. Next. Alors, au niveau des pertes maintenant d'acuité visuelle, donc il y a un patient qui est à part, c'est celui qui a une cataracte évolutive, mais on voit que par rapport au groupe témoin, on a une supériorité des pertes d'acuité visuelle, probablement due à l'évolution de l'atrophie ou à une dégradation des altérations de l'épithéliome pigmentaire et notamment des drusènes. Et donc, on voit qu'à chaque étape, la supériorité, le groupe témoin a plus de pertes d'acuité visuelle. Donc, ceci montre un résultat assez cohérent au niveau de l'acuité visuelle sur le gain, sur les pertes. Next. Alors, en termes d'acuité visuelle, on peut aussi tester l'acuité visuelle en faible luminance. C'est quelque chose qu'on fait peu en France. On va nous utiliser plutôt l'acuité visuelle, idéalement avec l'échelle ETDRS pour pouvoir avoir des gains d'acuité visuelle en lettres. Et donc, sur les faibles luminances, on voit qu'il n'y a pas de différence statistiquement significative. Néanmoins, on voit qu'il y a quand même une petite tendance. N'hésitez pas à préparer vos questions. Vous pouvez les mettre dès à présent dans la discussion et on se fera un plaisir avec Clark de répondre à toutes vos questions. Donc, quand les questions viennent, n'hésitez pas, vous les écrivez dans discussion en bas, vous cliquez sur Q&R et donc vous posez vos questions et on y répondra après. Next. Alors, au niveau du gain d'acuité visuelle, l'équipe Lumitera nous propose ici une comparaison assez intéressante parce que aujourd'hui, ce que nous propose la photobiomodulation, c'est le groupe vert, traité avec les 9 séances répétées tous les 4 mois, on a un gain au bout de cette année de traitement de 5,4 lettres en moyenne. Eh bien, si vous prenez sur le même laps de temps les patients qui ont été inclus dans l'étude Chroma et Spectri, on en a fait partie, donc il s'agit d'études ayant testé le lampalizumab, c'est un anticomplément dans la progression de l'atrophie, vous voyez qu'au même moment, vous avez une perte d'acuité visuelle. Donc ceci permet de mettre en balance le fait qu'on a un gain avec la photobiomodulation et qu'on est en train de traiter des patients qui ont potentiellement des risques de perte d'acuité visuelle liés à l'atrophie. Donc ceci est assez important et ça permet de mettre un petit peu l'accent sur ce traitement préventif, peut-être traité le plus tôt possible afin de protéger l'œil et d'assurer en fait l'acuité visuelle le plus longtemps possible. Next. Au niveau donc des premières études Lightsite 1 et 2, surtout l'étude Lightsite 1, on avait vu des gains au niveau donc de la fonction, l'acuité visuelle, et on avait également des gains au niveau de la sensibilité au contraste. Pareil, c'est un examen qu'on fait peu en France, en pratique courante, et je pense que c'est important d'avoir des examens qu'on va faire plus facilement en pratique courante pour ne pas changer nos habitudes. Et donc, on voit ici dans les deux groupes qu'il n'y a pas de différence statistiquement significative. Probablement que les patients ont été traités tôt et qu'il n'y avait pas encore d'altération de la sensibilité au contraste. C'est pour ça qu'on ne voit pas la différence. Next. Au niveau donc des critères anatomiques, donc je pense que c'est très important quand vous vous lancez dans la photobiomodulation d'avoir des critères fonctionnels et des critères anatomiques. Et vous avez des patients qui vont avoir une amélioration sur des critères anatomiques, d'autres qui vont avoir des améliorations sur les critères fonctionnels et d'autres sur les deux ou parfois aucun des deux. Et donc c'est important de monitorer vos patients pour pouvoir les suivre avec ces critères. Alors, au niveau du volume donc des drusènes, parce qu'on va s'adresser à des patients qui sont en DMLA intermédiaire, on voit qu'il n'y a pas de différence au niveau des deux groupes, entre les deux groupes. Il n'y a pas de différence. Néanmoins, ce qu'on peut observer au niveau numérique, c'est une augmentation du volume dans le groupe témoin. Vous regardez le volume jaune, alors qu'il y a une stabilité tout au long du suivi pour les patients verts traités par photobiomodulation. Pas de différence statistiquement significative, mais avec une tendance numérique qui est quand même à l'augmentation. Donc on a un peu l'impression que la photobiomodulation va agir ici en termes d'effet protecteur sur ces drusènes, donc on est vraiment dans la DMLA intermédiaire. Next. Il est intéressant de voir dans l'étude Lightsite qu'au niveau des changements à l'étude des 13 mois, c'est-à-dire les patients ont été traités donc tous les 4 mois, on voit qu'il n'y a pas de différence au niveau du volume, mais il y a quand même une augmentation, et vous voyez ici de manière très impressionnante la comparaison du volume des drusènes centraux. Et c'était une de mes premières constatations, c'est que la photobiomodulation agit très bien sur les drusènes séroconfluents, parfois même sur les DEP drusènoïdes, et donc il y a un effet quelque part protecteur sur le développement de ce volume de drusènes, et qu'on voit très bien marqué ici. Next. Alors, donc on a vu, l'efficacité fonctionnelle sur l'acuité visuelle à 13 mois. On a vu la différence au niveau des gains et des pertes d'acuité visuelle entre les deux groupes. On a une tendance au niveau anatomique. Et il y a également la question de la tolérance et de la sécurité du traitement. Alors, à mon sens, ce n'est plus un sujet parce que j'ai à mon actif une trentaine de patients, mais c'est toujours intéressant de voir sur ces études faites de manière très rigoureuse, de manière très bien faite, s'il y a où on déclare tous les effets secondaires possibles et imaginables, s'il y a un voyant éventuellement qui s'allume. Très clairement, aujourd'hui, il n'y a pas d'effets secondaires notables. Le patient tolère parfaitement. La question qu'on pouvait se poser également, c'est est-ce qu'il y avait éventuellement un taux de conversion plus important de DMLA néovasculaire? Pas du tout. On a même l'impression qu'il y a un moins de conversion au niveau DMLA néovasculaire, et c'est quelque chose qu'on avait déjà vu dans les études préliminaires Lightsite 2, c'est qu'il y a probablement un effet protecteur qui reste encore à démontrer sur le taux de conversion. Donc, il y aurait un effet fonctionnel et un effet protecteur. Au niveau des autres signes, pas tellement de choses à relever. Les patients peuvent se plaindre parfois d'un petit éblouissement qui va durer quelques minutes après le traitement, mais qui nécessite aucune précaution. Les patients viennent seuls à leur traitement. Il n'y a pas de dilatation et pas de gêne a priori. Next. Au niveau des effets systémiques, pareil, pas de signal, on agit par la lumière au niveau de l'œil, donc ce qu'on pensait est confirmé, il n'y a pas vraiment de retentissement au niveau général, et donc ce qui permet aujourd'hui, avec tous ces résultats de sécurité et de tolérance, de pouvoir utiliser ce traitement avec une sécurité totale et sans aucun arrière-pensée pour nos patients. Next. Donc en résumé, c'est aujourd'hui une chance de pouvoir avoir les résultats de Lightside 3 en avant-première. Il s'agit donc d'une étude américaine multicentrique avec une rigueur scientifique optimale, multicentrique, randomisée versus placebo, avec 100 patients sélectionnés dans les plus gros centres américains et suivis de manière optimale avec un protocole Lightside 3 qui est 9 séances de traitement répétées tous les 4 mois. Et donc, cette étude avait pour but de montrer le gain d'acuité visuelle, et donc cette étude a montré effectivement de manière statistiquement significative le gain d'acuité visuelle au bout d'une année de traitement. Cette étude a permis également de montrer que le gain était ceux des études précédentes entre 5 et 6 lettres retrouvés, avec parfois des patients qui avaient des gains de plus de 15 lettres. Il n'y avait pas de différence au niveau de l'acuité visuelle en faible luminance, pas de différence au niveau de la sensibilité au contraste. Il y avait une tendance au niveau des volumes sans avoir de différence statistiquement significative, néanmoins avec une progression du volume des drusènes pour le groupe témoin. Il y avait une différence statistiquement significative pour l'acuité visuelle corrélée aux drusènes, et donc c'est probablement une des voies d'action de la photobiomodulation sur le gain d'acuité visuelle. Donc, un mot sur la tolérance, aucun effet secondaire relevé, et donc ce qui permet de réaliser la photobiomodulation dans les conditions optimales. Donc, en fait, on retrouve avec l'étude Lightside 3 ce qu'on pensait déjà avec l'étude Lightside 1 et 2, on retrouve les mêmes tendances, et je pense que ce qui est intéressant, c'est de discuter, de pouvoir échanger tous ensemble. Donc, n'hésitez pas à poser vos questions, on est là pour répondre tous ensemble, pour appréhender et surtout pouvoir proposer à nos patients ce traitement totalement innovant. Encore merci à toute l'équipe de l'Umitera, merci pour votre attention et on est là pour vos questions.
Mathé, il y a quelques questions. Clark, there are a few questions, okay? Première question, first question. Qu'est-ce qu'on fait si les patients ont des cataractes? Est-ce que ceci a un effet sur le traitement? What if the patient has cataracts? Does it have any effect on the treatment? I think a question for both. C'est une question à vous deux.
Would you like me to start, Attila, on that one?
As you like.
So in the clinical trial, obviously cataract progression is a concern because it will impact your ability to continue to take BCVA or other contrast sensitivity tests. We typically have enrollment exclusion criteria. If you have an early cataract and the investigator believes that it will not progress over the course of 2 years, they can enroll the patient. But the majority of the patients are post-cataract. In terms of the wavelengths, it will get through the cataract, but obviously we had one patient in here that had significant vision loss. It was a PBM treated one with a cataract that did, over the course of a 1.5 to 2 years in the trial, continue to lose vision. So from a clinical trial standpoint, it is important to eliminate those. We will stratify a separate analysis without the cataract causing vision loss. But from a standpoint of treatment, the wavelengths can get through the cloudiness of the lens.
Moi, effectivement, sur la cataracte, c'est une question très intéressante parce que on a nos patients qui sont à l'âge de la cataracte. Et donc, ce qui est très important, quand vous allez réaliser la photobiomodulation, vous allez voir qu'il y a une partie où le patient a l'œil fermé. Donc, il s'agit de longueurs d'onde qui vont agir à travers la paupière et donc qui vont aussi agir à travers la cataracte. Donc, à mon sens, le seul risque, c'est qu'éventuellement la lumière soit un tout petit peu bloquée, mais regardez dans l'étude Lightside 3, même dans l'étude Sham où il y avait 100 fois moins d'effets, il y avait quand même un tout petit effet. Donc je pense que la cataracte n'est pas du tout un problème, et quand on a une indication, eh bien il faut le proposer, soit pour des patients pseudophaques, pour des patients cataractes, mais peut-être qu'on peut avoir la pleine efficacité du traitement sur des patients pseudophaques. En tout cas, s'il y a une autre question, est-ce que la photobiomodulation aggrave la cataracte? Clairement, non.
Mathé, il y a une question pour toi de l'audience. Tu peux le lire?
No problem. Oh, this is an interesting question I have from the audience, and it was a many discussion with Clark concerning this question. Clark, can you give us your thoughts? The question is, what are the limits for visual acuity for patients that we want to treat with PBM? What do you think about the limitation, and what would be the good visual acuity to be treated with PBM?
Right. It's a great discussion. And, you know, our early studies casted a broad net from patients that were 20/40 to 2200. And what we saw was that there is an anatomical correlation between the amount of central GA and your ability to respond to PBM. However, even those category 4 patients that have islands of viable tissues, about 25% of them can respond very well, and sometimes it will surprise you. I think the question for the clinicians to the patients is a different one. It's not so much can we gain 5-10 letters back, but can we slow down the progression and keep what you have and maintain you know, whatever visual acuity you have. So we do see, you know, our doctors in Europe and South America that are treating all stages. We think that earlier where you have viable tissue, and I think the slide that Matea showed that shows the GA study, clearly these patients are a little bit earlier than those patients that had either unilateral or bilateral GA. You can see once you get to that point, we can't fix it, and your progression is very rapid within about 5 letters in 48 weeks from 2 very large studies. So we believe if you treat earlier, that is better. Earlier you get, and this is the beauty of the eye, that we have the ability to use imaging and other biomarkers to see the disease early. So we actually have a window to treat these patients before we have irreversible GA and can't fix what's gone.
Alors moi, je vais compléter aussi sur mon expérience. Donc moi, j'ai commencé par vraiment traiter des DMLA atrophiques avancés, et donc sur des acuités visuelles très faibles, très faibles. Et en fait, j'étais un petit peu déçu sur le gain d'acuité visuelle, parce que probablement qu'au niveau des photorécepteurs, c'était déjà un petit peu tard. Donc ma tendance au fur et à mesure de l'expérience Valeda, ça a été de traiter de plus en plus tôt. Et donc je pense, pour en avoir discuté beaucoup avec Clark, c'est la bonne indication, c'est autour de 6-7 dixièmes. Parce qu'on commence déjà à avoir une petite altération et le patient va sentir l'amélioration. Parce que si vous traitez un patient à 10 dixièmes, bien sûr, vous n'aurez pas de gain d'acuité visuelle. Donc, comment juger le gain? Et donc, en fait, la bonne indication pour essayer de trouver l'efficacité, c'est autour de 6-7 dixièmes. Néanmoins, on peut avoir des patients au niveau de la qualité de vision qui peuvent aussi avoir des améliorations, le ressenti du patient. Et puis après, c'est des choses qui ont tendance à évoluer. Ce qu'on voit avec la photobiomodulation, c'est qu'on est parti de traitements avec des DMLA atrophiques évolués, et on est en train de s'orienter quand même sur du traitement d'une DMLA intermédiaire, ce qu'en France on appelle une MLA. C'est la maculopathie liée à l'âge, définie par des drusènes séreux, donc supérieurs à 125 microns, et avec des altérations de l'épithélome pigmentaire type hypo-hyperpigmentation. Donc c'est ça, les patients, et on va pouvoir donc les traiter quand ils ont une petite baisse d'acuité visuelle. C'est vrai qu'on a des histoires sur des patients qui ont des récupérations d'acuité visuelle. Moi, je l'ai eu ça en consultation, la patiente qui ne conduisait plus trop parce qu'elle ne se sentait pas à l'aise et après les 9 séances de photobiomodulation, a eu un gain d'acuité visuelle sur les 2 yeux. C'est une de mes patientes qui a eu 20 lettres de récupération sur chaque œil et donc qui a repris sa voiture et une vie normale. Donc l'acuité visuelle, c'est une bonne question, mais probablement pas facile à trancher. En tout cas, notre ressenti, c'est plutôt ça. Et vous allez voir qu'il y aura d'autres critères qui vont rentrer en ligne de compte. L'étude Electrolyte nous avait parlé d'électrophysiologie qui peut être intéressant pour cette discussion également.
That goes maybe in line with the next question. C'est pareil pour la question suivante. Il y a des patients avec des drusnes, mais encore une bonne acuité visuelle. Est-ce que vous traiteriez ces patients? Another question which goes in line with the first question, Clark. There are patients who have drusen but still have a good visual acuity. Would you treat those patients, maybe preventively? Question
Yeah,
for both.
again, that extends the concept of what drusen really is. I think drusen is showing a correlation between BCVA in our treatment patients. However, my sense is drusen is more of a surrogate for cellular health. It's the hallmark pathology. It's indicating what's to come, that the cells are sick, that they're no longer able to remove the lipofuscin products and they're depositing. And so then you create an inflammatory nidus, ischemia. And so, yes, I think that once you have signs of the disease, if you start to treat, what we're seeing is if the cells are healthier, you're no longer seeing additional deposition. And the pathways out there to remove that ultimately can improve the overall pathology. So I do believe that what we're looking at here is an opportunity to treat those patients before you start to have visual loss and obviously irreversible geographic atrophy.
C'est une bonne question. Moi, je pense qu'il faut d'abord se poser la question, qu'est-ce qu'on recherche avec le traitement? Est-ce qu'on recherche un gain d'acuité visuelle? Est-ce qu'on recherche une protection sur le court, moyen, long terme? Et donc, probablement que si vous cherchez un gain d'acuité visuelle, bon, traiter un patient à 10 dixièmes, vous n'aurez pas grand-chose, mais que si vous avez des drusènes, typiquement, c'est le genre de patient. Je vais vous dire le patient qui serait parfait, c'est le patient qui a des drusènes séreux confluents, qui évolue et qui a déjà de l'atrophie d'un côté. Il a fait une atrophie centrale. Ce patient, il veut tout faire pour éviter que ça évolue. C'est la vraie place. Traitez-le le plus tôt possible avec les compléments alimentaires et avec de la photobiomodulation. Pour moi, un des clés, ça a été l'étude Electrolyte. On a eu la chance de faire une webconférence avec l'équipe Lumitera là-dessus, où on a vu que de manière électrophysiologique, il y a une efficacité de la photobiomodulation et probablement que l'acuité visuelle, c'est un bon indicateur, mais également l'électrophysiologie, parce qu'on a des critères, notamment au niveau de l'ERG, qui permettent de voir un dysfonctionnement, et quand vous les traitez avec la photobiomodulation, ça permet de remettre la fonction électrique. Ensuite, sur la protection à moyen long terme, c'est ce qui a été soulevé dans les premières études Lightsight 2, c'est si vous regardez le taux de conversion vers l'atrophie ou des taux de conversion, il semblerait y avoir un effet protecteur. Donc, dans ces cas-là, vous allez plutôt utiliser la photobiomodulation pour protéger sur le long terme votre patient.
Taya, I would give one additional example. In the Lightsight 3 study, or sorry, Lightsight 1 study, we had a patient that was 52 years of age that had significant central drusen, had come in because of blurred vision, just starting to see some impact on contrast sensitivity and BCVA. By about one round of treatment in 7 months, it had reduced itself to about 85%. And by a year, with no further treatments, it had resolved. So when you're talking about an early patient that may be just at that cusp of seeing some vision impairment, we were able to, a year later after one treatment, there were no signs of disease. No indication, that we cured him because it could come back, but you were able to make a significant pathological improvement with one round of treatment because we caught it early.
We have a nice question about is there any
Vous pouvez, Mathé, tu peux lire les questions?
yes, on a une question que je trouve très intéressante, c'est y a-t-il une acuité visuelle en dessous de laquelle le traitement n'est plus efficace? Clark, there is one question, is there a visual acuity below we will not use photobiomodulation because we are sure there is no any efficiency. And I think it will be interesting to share your experience in Finland, or was it in Scandinavia?
Norway.
In Norway, yes. I think this is a very interesting case.
There was a case, we have a retinal center in Norway that's now probably treated in excess of 200 or 300 patients. One of the cases that she pointed out was someone that came in that had a significant amount of geographic atrophy. She was really not particularly interested in treating that patient, but they were obviously, had a significant vision loss. Retired gentleman that liked to hunt and go to the gun range and shoot. And his left eye could no longer do that because his left eye was impacted. They treated, and the patient gained significant vision, was able to go out and do some of the activities in his retirement that he loved to do. And quite honestly, the investigator was surprised because of the amount of GA. So I think it goes to the random growth of the lesions. There still could be viable islands of tissue that is contributing to the potential to either give you back some vision or improve what you have. And so I think, I look at those cases and what they did to the impact of the quality of life of that patient. And that's important, obviously, for the patient.
Une autre question pour les PDR et les migrations pigmentaires. We have a question about the different phenotypes of intermediate AMD. Shall we look at these different types? Is there any recommendation?
Well, I would say, we're still just looking at the top line data, so we will have additional cuts. One of the things that we haven't seen in this in LightSight 2 was a big impact in contrast sensitivity, because the patients actually had pretty good vision and were using the Mars contrast sensitivity, which isn't as sensitive as the FACS CS scale. In terms of pathology, we want to look at how we're correlating to improvement in stage of disease. And we've seen that earlier in a broader swipe that those patients that had central fovea GA involvement didn't respond as well as you might expect. But it would be interesting to see if there is hyperfluorescence, if there are other aspects that we can identify patients that either will respond better or that won't respond. There are other metabolic markers. One of them that we've done is electroretinogram. And what we've seen is improvement in retinal ganglion function using the full field flicker or multiluminance approach. That tends to be early and more quantitative. So we like that. Microperimetry with patients that have fixation stability issues. We've seen several of our European doctors use microperimetry and show improvements in fixation stability. So there's definitely a lot of work to understand the different stages of pathology and how we can optimize treatment. So I'm fully supportive and agnostic to imaging. Biomarkers like ERG, I think, are very interesting. You might be able to identify when the treatment needs to be redone. And so that could be coupled with the Valeda treatment. So I think there's a lot more research to do as we expand our knowledge on PBM and how to make it even better.
Alors, moi, je voudrais, pareil, la question sur les différents phénotypes, je me suis posé dès le début. Et donc, en fait, globalement, dans mon expérience, excellent résultat sur les drusènes séreux confluents. Sur le matériel, dystrophie pseudo-vitelliforme matérielle, j'ai pas constaté d'efficacité. Et sur les pseudodrusènes réticulés, je n'avais pas l'expérience. Mais la question est bonne parce qu'on sait que ça évolue souvent soit vers de l'atrophie, soit vers des phénotypes particuliers de DMLA. Et donc, le professeur Souilled, Éric Souilled, à Créteil, a lancé une étude rigoureuse sur les différents phénotypes et il a présenté à la SFO les premiers résultats donc de l'étude. Et c'est intéressant parce qu'il regarde la réponse à la photobiomodulation en fonction des différents phénotypes. Et donc, on apprend énormément de ces résultats. Et la conclusion qu'il nous a présentée à la SFO était de dire qu'il semblerait y avoir un résultat sur les drusènes séreux, les drusènes séreux confluents, mais également sur les pseudodrusènes réticulés. Donc ça, c'est très intéressant, peut-être sur le nombre de pseudodrusènes réticulés qui semblerait diminuer. Sur le matériel, il semble ne pas retrouver d'effet. Et ce qui est intéressant, c'est qu'il va regarder le terrain génétique par rapport aux effets de la photobiomodulation. Il y a également une discussion très intéressante à la Société française de rétine sur la photobiomodulation et il y avait le professeur Giuseppe Carques qui était présent de Milan, un ancien de Créteil qui est maintenant professeur en Italie et qui regarde aussi dans les différents phénotypes. Donc c'est des questions très pertinentes et on aura les réponses assez rapidement. En ce qui me concerne, plutôt les drusènes séreux confluents.
Il y a une question concernant les suppléments ARETS. There is a question about the ARETS supplements. Recommandez-vous de prendre des suppléments ARETS pendant et après le traitement? Partiellement, vous avez déjà répondu. Clark, the question is, do you recommend to take ARETS supplement during and after the treatment? It was partially answered already, but could you please comment?
Yeah, I mean, I think that in the US versus Europe, we see a much higher use of AREDS vitamins within the patient population. In our first study in Canada, it was about 100%, 86% in the Light Sight 3, and only about 30% in the European Light Sight 2. We did look at those patients that had greater than 5-letter response, and it turned out none of those were on the AREDS vitamin. It was a small set. We are stratifying Light Sight 3, so we'll have more data to determine if there's any benefits. But generally, and you know, I'm looking at it as a different approach. They're antioxidants. They have some benefit. They've been shown to slow the progression in about 20–25% in the AREDS studies. So I do think there is a benefit to have healthier tissue, and antioxidants in terms of AREDS vitamins seem to be doing that in a portion of the population. So I would you know, recommend that it doesn't have to be just one treatment. You know, there's no magic bullet here. And if we can have some incremental benefit on other pathways with PBM or other treatments, that's the way that we probably need to approach these degenerative diseases.
Pour moi, c'est clairement un grand oui. Il s'agit de l'indication des vitamines AREDS et probablement qu'il y a un effet potentialisant entre les deux, donc un grand oui.
Ok, merci les deux. Finalement, une question très intéressante pour le futur. Est-ce qu'il y a d'autres indications cliniques à part de la DMLA atrophique et drusen? Clark, a question maybe also for you. Okay, directed to the future. Do you see any other clinical applications apart dry AMD and drusen?
Yeah, that's a question that usually comes around, is that where else could we use this? And there obviously are acute and chronic inflammatory disorders. We like, initially, we have some work in one of our centers in Germany in diabetic retinopathy patients with macular edema. These are early patients that have pretty good vision, and those are the ones that you're watching before you start with the anti-VEGF injections. So there's a potential there. There's a potential that in AMD, and the anti-VEGF drugs are very powerful, very good, revolutionary in ophthalmology. But the burden of injections is significant, and we know that they're being undertreated. So PBM to maybe minimize the time between anti-VEGF injections. So it could be either additive or synergistic would be interesting areas of research. There's other things, central serous, other maculopathies that some of our doctors are showing interest in moving into. Stargardt's would be another interesting opportunity of the earlier genetic disorders as well. So I do think this is a platform. Each disease is going to be a little bit different, and we're going to have to do trials to demonstrate its efficacy. So we're clearly looking at this as an opportunity to treat more than just dry AMD.
Effectivement, je pense qu'au niveau des indications, donc la DMLA intermédiaire, atrophique est la porte d'entrée, mais que les indications sont nombreuses. Moi, je serais intéressé par la CRSC, parce qu'on sait aujourd'hui qu'on n'a plus de PDT, on n'a rien à proposer. Moi, je trouve qu'il y a une vraie place éventuellement pour tout ce qui est traitement de la pachychoroïde. Donc ça serait des choses à évaluer, l'œdème maculaire. Et je suis intimement persuadé qu'à l'avenir, dans X temps, on aura des traitements combinés dans la DMLA avec les injections d'anti-VEGF et la photobiomodulation, parce que vous savez très bien que 80% des patients passés 5 ans ont de l'atrophie dans la DMLA néovasculaire, et c'est ce qui va limiter leur acuité visuelle. Donc si on a un traitement qui traite la composante néovasculaire et qu'en plus on peut protéger de l'atrophie, eh bien on peut maintenir une acuité visuelle optimale. Donc ça, ce sont des rêves, ce sont des pressentiments, et bien sûr, voilà, c'est des choses qu'il faudra vérifier et démontrer par la suite.
Intéressant, Mathieu. Oui, très, très intéressant. Finalement, une question que Clark a déjà brièvement répondu, mais c'est venu de nouveau. Finally, one question which you, Clark, already answered a little bit, but it came again. Est-ce qu'il y a une corrélation entre les drusen et les potentiels gains de vision? Clark, the question which you have answered briefly is there a correlation between drusen volume and potential gain of vision?
So Attila, it's a little bit of a complicated answer. In some cases where you have central drusen and it may be distorting vision, those patients, we do see improvement in vision, contrast sensitivity, or BCVA with reductions in drusen. In the earlier stage patients where there's smaller amounts of drusen, we're seeing in our European and our US trials no drusen deposition. So while we're getting improvement in vision, drusen is no longer being deposited. In the sham, where we're getting minimal improvements in vision, drusen is continuing to get bigger. So there is a negative correlation there. It's not very strong because, as you'd expect, it's not a one-to-one correlation. It's a surrogate for health. It's continuing to get worse. And there are data that suggest the more drusen you have, the quicker the disease progresses. So I think it's complex, but there is an association there.
Okay. Mathieu, tu voudrais ajouter quelque chose?
Oui, moi je trouve ce qui est difficile, ce qu'il faut bien comprendre pour nos auditeurs, c'est que la photobiomodulation, c'est le savant dosage d'une longueur d'onde et de l'exposition à cette longueur d'onde. Et donc en fait, quand on traite avec des médicaments, on va donner, ou des compléments alimentaires, on va donner une molécule. La photobiomodulation, eh bien, c'est une longueur d'onde délivrée avec une certaine durée et certains intervalles. Et c'est ça qui fait toute la différence. Et c'est ça la difficulté, c'est qu'il faut trouver la bonne indication avec le bon schéma thérapeutique. Et probablement, c'est là-dedans où il faudra s'améliorer sur certaines indications, peut-être faire 9 séances, sur peut-être certains en faire que 6. Donc, en fait, c'est de trouver le bon schéma thérapeutique pour répondre à cette indication. Et c'est ça qui est difficile. Probablement que certains sceptiques de la photobiomodulation sont originaires de tentatives de photobiomodulation qui ne sont pas les bonnes longueurs d'onde et pas les bonnes expositions. Ce qui m'a séduit donc avec l'appareil Valeda, c'est qu'en fait la longueur d'onde est contrôlée en permanence pendant l'émission, et donc en fait il y a une sécurité, l'appareil se calibre en permanence pour toujours émettre à la bonne longueur d'onde voulue.
Ok, merci. Finalement, une question pratique. Est-ce que ceci pourrait être utilisé par un technicien ou un infirmière? One practical question is, can it be used also by a technician or a nurse?
And the simple answer is yes. We have set the device up so all 3 wavelengths in the protocol are based on our clinical trial data. They're preset, so it can be used by a trained technician, nurse in the hospital or private clinical setting.
Je rejoins tout à fait le Clark, et je pense que je pourrais le faire aussi par ma fille. Donc, alors, elle est très forte, c'est ma fille, donc, mais bon, elle a 9 ans. Alors, nous, notre expérience au centre Export Vision, c'est uniquement des médecins qui font les 9 séances, tout simplement parce que le protocole est nouveau. Pour l'instant, il y a l'expérience à connaître. Donc, toute l'équipe d'Export Vision s'y est collée et tout le monde fait les séances à tour de rôle. Ça demande du temps, de l'investissement. Mon impression, quand on regarde les protocoles, quand on regarde les critères de sécurité, c'est qu'en fait, clairement, il s'agit d'un traitement délégable, clairement. L'appareil est très facile d'utilisation et on a un écran qui fait l'interface, et cet écran explique. Donc, c'est-à-dire, il faut vraiment voir que vous avez ce qu'il y a à dire écrit sur l'écran. C'est-à-dire, il y a marqué, vous pouvez fermer l'œil. Une fois qu'il a fermé l'œil, on clique sur valider. Donc, même si vous ne connaissez pas encore le process que vous découvrez, il suffit juste de lire ce qu'il y a sur l'écran, et l'appareil est intelligent parce que si le patient n'est pas bien positionné ou qu'il bouge la tête, le traitement s'arrête. Donc, vous repositionnez la tête et il recontinue de là où vous avez arrêté. Donc, je pense qu'il y a une sécurité optimale et clairement délégable. Oui.
Finalement, une autre question est venue. Y a-t-il une amélioration des ERG dans les études faites? Question Clark, which just came. Is there an improvement of ERG during the studies? It refers to
Yes.
the ERG study. Yeah.
We conducted a study that finished last year in the US using the diopsis ERG. It's called the electrolyte study, and we've presented that. We can provide abstracts for the study. The design of the study was to measure baseline ERG and ERG prior to each week's visit. And what we saw over the course of 3 weeks of visit, where the patients came in typically Monday, Wednesday, Friday, was we got an increase in multiluminance area under the curve, the magnitude of their response at the retinal ganglion level. We saw an improvement in different protocols and fixed luminance and negative atomic responses. And what that indicated to us was that over the course of the 3 weeks of treatments, we were getting improvement in retinal ganglion function. It was interesting that just under 50% of the patients maxed out around 2 weeks. And then we followed the patients out for 3 and 6 month follow-ups. And what we saw with these same patient intermediate patients is that we were getting an improvement in BCVA, about 14 letters, improvement in Mars contrast sensitivity at 3 different distances. And they were maintaining it out to about 6 months when we started to see the ERG response diminish. And so we do believe it can be used to optimize not only treatments, but potentially could be used to find out when the patient needs to come in for the next round of treatment. So I do believe biomarkers are going to be very useful in optimizing your patient and even within the patient, you know, the treatment to that eye.
Alors, pour répondre sur la partie électrophysiologie, moi, je pense que c'est capital. Voilà, c'est capital. Et donc, on a les critères faciles, fonctionnels anatomiques, acuité visuelle de TDRS, OCT pour le volume et l'anatomie. Mais je pense qu'en termes de fonctionnel, l'ERG avec la multiluminance flicker est le critère principal. Et donc, aujourd'hui, maintenant, je l'utilise pour savoir s'il y a une bonne indication et je l'utilise pour voir la réponse thérapeutique. Et je pense que cet ERG doit être réalisé avec l'appareil Diopsis, qui est un ERG nouvelle génération, où en fait vous n'avez pas un électrode cornéen, mais vous avez un électrode qui est collé sur la paupière et qui permet d'extraire en fait le résultat depuis l'enregistrement de ces électrodes palpébrales. Donc c'est un appareil, enfin voilà, on a été les premiers à l'utiliser, donc nous on l'utilise en pratique courante aujourd'hui pour tout ce qui est antipaludéen de synthèse, bilan des rétinopathies pigmentaires et autres, mais donc je l'utilise aujourd'hui maintenant pour les bilans pré et après traitement par Valeda, donc je pense que c'est un critère majeur.
Merci les deux. Finalement, une question très intéressante pour toi, Mathé. Docteur Mathé, avez-vous essayé d'autres appareils de photobiomodulation concurrents? Merci. Very nice question. Maybe Clark, you can also comment. It was directed to Mathé. Mr. Mathé, have you used other devices for photobiomodulation from opposition? Thank you.
Alors moi, je vais répondre facilement et je pense que Clark va développer parce qu'il connaît plus. I will answer quickly because I don't have any other experience. But I think it will be interesting, Clark, that you share with us your thoughts about other devices. We discussed about it. Alors moi, pour répondre facilement, non, parce que depuis que je regarde, à ma connaissance, il n'y a pas d'autres appareils de photobiomodulation avec marquage CE disponible en Europe. Il y a eu récemment des études sur certains types de photobiomodulation qui n'ont pas montré d'efficacité, mais c'est tout l'intérêt, à mon avis, du dispositif Valeda, c'est que tout dépend de la longueur d'onde et du sérieux qui est fait autour. Clark, give us your thoughts.
Well, I appreciate the question, and you know, I think what's unique about the Valeda is it is a multiple wavelength. We've chosen each wavelength based on the underlying science around it, and feel that that's important. That some of the single wavelength, the LEDs, are not addressing particularly the targets, both of the substrates, and do not have the background science that we've put into the Valeda. We've done cadaver tissues absorption scatter studies, beam profiling, phantom eye, to create a unique expanding bundle of light so that we can identify the spot size at the retina, both with the open eye and closed eye. There's a lot that's gone into developing that. So some of the other, let's say, other instruments out there have not gone to the level to create an eye-safe instrument for the doctors where we fully homogenize the beam. We also have run more clinical trials than anybody else. So I think there's value in somebody that is putting, you know, their efforts into working with the doctors to create a technology that could be a tremendous value for the patient. So I'm biased, obviously. I like the Valeda, and I hope you guys do as well if you try it.
Merci les deux. Les temps passent si vite. On est déjà 8 heures. Time flies very fast. It's already 8 o'clock. Okay. Nous remercions aux orateurs pour les excellentes présentations et aussi pour votre participation active. Si vous avez des questions supplémentaires, n'hésitez pas à nous contacter. Vous pouvez voir nos courriers électroniques. Vous pouvez aussi contacter notre représentant en France, Monsieur Régis François Gansel. Et finalement, nous vous souhaitons une bonne soirée et surtout, restez en bonne santé. Bonsoir à tout le monde.
Matej, thank you. Thank you all.
Merci Clark. À très bientôt.
Good afternoon for those on our side of the pond. We just are very excited to put on this webinar for you this afternoon, and we would like to welcome our speakers. We have a pretty full agenda, and the faculty for today, I'd like to introduce Professor Ben Burton. Ben, if you don't know him, was trained at Cambridge University and Moorfields Eye Hospital and was appointed to set up a age-related macular degeneration service at the James Paget University Hospital in 2007. He's a research and development director for the Trust and has built up a large retina trial unit with the state-of-the-art imaging equipment running between 4 to 12 trials at any one time. And his research projects include home monitoring and early detection of wet AMD and with AI, and also his role in the gut micromet in dementia and AMD, and he's the chief investigator for the UK for LightSight 2 trial of photobiomodulation in dry AMD, as well as being a PI for many other industry studies for age-related macular degeneration and diabetic retinopathy. So we welcome Ben to the faculty, and we also welcome Clark Tedford. Dr. Tedford is the co-founder of Lumathera and has served as president and CEO since the formation of the company in 2013. During Dr. Tedford's 30 years in the pharmaceutical and medical device industry, he's held scientific and executive management positions at Omeris, Solentex, Gliatech, and Schering-Plough. Clark earned his PhD in pharmacology at the University of Iowa and then completed his postdoctoral work in the Department of Pharmacology at Loyola University Medical School in Chicago. So we welcome you both. We are very pleased to present to you today our latest data, and as well as some updates and some information regarding photobiomodulation that I think you'll find very interesting at the least, exciting at the most, and we thank you for being here for the next hour with us. So without further ado, we will get started, and Clark, we'll let you lead off.
Thank you, Dwayne. And Ben, it's a pleasure to be able to speak again with you today on our progress. The agenda for today is I'm going to spend a very short period of time talking a little bit about the mechanism of actions on photobiomodulation and really the Valeda multiple wavelength PBM approach. We're going to turn it over to Ben, who can talk to the LightSight 2 European post-marketing study that he was one of the PIs in a multi-center trial. And then today, following that, we're going to compare that and bring up the new data that we have from the US pivotal trial, the LightSight 3. We've got top-line data. We're still obtaining more information as we go through, but it gives us an opportunity to compare and contrast what we've seen between the two studies and as we continue to build this out. This is an interactive program, and there is a Q&A box. We would like you, if you have any questions, just to go ahead and submit them throughout the time of the webinars today, and we'll try to finish early so we can have some discussion around the Q&As. So with that, I'm going to get started here. As Dwayne had indicated, I am one of the founders. Lumathera was founded in 2013. We're in the Seattle, Washington area. I'm the lone pharmacologist. We had a couple engineers and 2 eye doctors to form the company. And our goal was really to create a non-invasive treatment for degenerative eye disease using photobiomodulation to hopefully improve visual function, slow or stop progression of these diseases, and hopefully prevent vision loss and blindness. And with that being said, The technology we're using is photobiomodulation. It's also known as low-level light therapy. It's a very exciting medical technology that there's a lot of research in various areas that are continuing to expand its potential and allowing us to understand the core mechanisms. It really is using low levels of light to stimulate cellular function. Really specific targets can be chosen based on the wavelengths, and we typically use visible to near infrared wavelengths. Right now, it's approved or being used in physiotherapy by particularly in the skeletal muscle area, in arthritis to reduce inflammation and pain. In diabetics, some examples of stimulating and closing wounds have made great progress. In sports medicine, athletes are using it if they tweak their knees or ankles to help improve recovery and get them back into top shape. In other areas, in dentists, in oral cancer, prevent against radiation damage, and in particular, what we're going to talk about today is in retinal disease. And so the mechanisms that we're targeting in photobiomodulation is 3 different wavelengths. 2 of them target an enzyme. In the cartoon on the left shows a depiction of cytochrome C oxidase, which is a mitochondrial target. It has 2 spots that accept light. The 2 wavelengths are 660 or 670 in the far red that really stimulate the iron-copper binding site for oxygen, where the reaction takes place in reducing oxygen to water and pumping protons across the intermitochondrial membrane. The other one is 850, which is near infrared, and it's involved in accepting photons at copper A, which helps shuttle electrons in. And so both electrons and oxygen are key substrates for this regeneration or re-stimulation of the proton pump. That gradient leads to the next enzyme, ATP synthase, creating stored energy. And so as we age, particularly high energy neurons or retinal cells, they tend to be susceptible to reductions in mitochondrial function. Ultimately, if the cell cannot make enough energy, it will trigger apoptosis and cell death. The cytochrome C oxidase is a key 4th of the 5 enzymes involved in electron transport and really can regulate and set metabolic activity. So the Valeda Light Delivery System was chosen and created using these 3 different wavelengths. The 3rd one is actually used to reduce VEGF expression and potentially could be used to augment some of the benefits in the wet AMD area. Our focus has been really on the dry form, though, and the product has been CE marked and available in Europe for about 2.5 years. We have been continuing to build to our clinical trial data over the course of the last 3 years. And this just gives you an example. In the last year, we had 3 studies. We're going to talk about the European Light Sight 2 trial that Ben participated in. But we also had a study in early DME patients with good vision that came out and was presented at ARVO last year. We've done a biomarker study with electroretinogram demonstrating improvements in ERG and retinal ganglion function over the course of treatments. And most importantly, the most recent study is our pivotal Light Sight 3 trial, which is similar to the 2-year trials with Eylea or Lucentis. It has a 13-month time point. We've now got the top-line data from the 13-month efficacy time point, and we'll share that today with the group here. So with that, I'm going to leave this over to Ben so he can give kind of a background on the previous study, Light Sight 2, that was conducted across Europe. And Ben, with that, I'll run the deck and introduce you and let you take off.
Okay, thanks very much, Clark. Thank you for inviting me. So I was the chief investigator in the UK for Light Sight 2 trial. It's a European trial. You can see the centres here. That we had a couple of 3 centres in Germany, a couple in the UK, Italy, France, and Spain. And it was a double-masked, placebo-controlled, sham-controlled trial, multi-centre, looking at photobiomodulation in really intermediate dry AMD. Next slide. So this is the award-winning Valeda system, which is, as it says, user-friendly, won lots of awards and sort of impressive piece of kit. Next slide, please. We'll look at that in a bit. What this does, it's all pre-programmed, so there's just a couple of buttons on it. You have to press, just line the patient up, and then the treatments are given blocks of treatment, and so they're very quick treatment episodes, about 5 minutes, and you do that 3 times a week for 3 weeks. Usually you can spread it over a bit longer. So one block of treatment is 9 visits for 5 minutes each. And it's, if you have a go on one of these, you don't need to be dilated. You just sit there, eyes open, eyes closed, eyes open again, and as a machine will tell you what to do. And it's basically kind of like looking into a sort of psychedelic sun. It's a very interesting experience, which you should have a go at if you ever get a chance. Next slide, please. So it's adjustable tabletop, so it's easy to sort of fit in the office, very user friendly, and really pretty straightforward. Next slide. So the Light Sight 2 design, the inclusion and exclusion, obviously you had to have AMD, so had to be over 50 by definition. And the vision, although it could be quite poor, and some of the patients had geographic atrophy, not all of them did, some of them just had a few drusen, so intermediate AMD, and some of them had really quite good vision, not very many having very poor vision from that. You couldn't have center involving geographic atrophy, so we're not, this wasn't proposed as a treatment for people who've kind of got count fingers vision and with loss of the fovea and trying to regain sight. That's not expected to happen. So the proposal was to have 96 subjects, 144 eyes. Unfortunately, as you'll all be aware, COVID happened right in the middle of our trial. So we'd got a few patients in, and it caused a certain amount of havoc with some patients wanted to carry on, some patients were too frightened of COVID to come in. And so it did make the whole trial a bit truncated, was a shame. It was randomized 2 to 1, so twice as many people got treatment compared to the sham treatment. And each block of treatment, each block of 9 light treatments was repeated every 4 months rather than every 6 months, which was the Light Sight 1 trial that came before this with a smaller number of patients. Next slide, please. So here we go, the baseline, the treatment. So we saw the patients a lot for about 3 weeks in a row, 3 times a week, so Monday, Wednesday, Friday usually, for 3 weeks in a row. Then we didn't really see them for about 3 months. And then they came back and had another intensive treatment, seeing them every day, every couple of days for 3 weeks in a row. And so 3 blocks of treatment a year, and then we assess them at 10 months so that the effective treatment wouldn't have completely worn off, but also just to give it time to work as well. So that was a plan. Next slide. And so the primary endpoint was best corrected visual acuity, but also some interesting secondary endpoints. In fact, probably more interesting to me, particularly draw attention to the drusen volume and thickness as one of the secondary endpoints, but also the geographic atrophy growth, one of the exploratory ones there, and also obviously looking at safety and adverse events. Next slide, please. So this is just to give you a rough idea of the distribution. So we had quite a few patients at our sites. We've done quite a lot of research, and we're getting a lot of patients, particularly those that were being seen regularly with wet AMD that we knew were reliable attenders. So we were just treating one eye in the study. Last visit was December 2020, and we're currently on the point of submitting the paper for publication coming from this now. Next slide, please. So 8 European centres. In the end, we ended up with 44 patients, 32 photobiomodulation treated eyes, 19 sham eyes, intermediate AMD patients. So some had geographic atrophy, but by no means all. And primary analysis was the vision, but multiple other OCT-based analyses as well. Next slide, please. 100% Caucasian patients. I work in an area where it's predominantly Caucasian, so not too surprising there. More females than males, again, not surprising because women tend to live longer and therefore more likely to end up with AMD, age-related condition. Mean time since diagnosis of AMD is about 3.7 years, so still quite early. Normally with dry AMD, the sort of time to blindness is roughly 10 years, people that go blind, and 74-year-old patients, so pretty typical, I think. Next slide, please. So the visions at baseline were pretty good. And in fact, one of the difficulties of recruiting patients into the study was the vision having to drop to below 6/9.5, even though they had drusen. So vision better than 70 letters is basically driving standard vision, 6/12, if you're not familiar with the TDRS letter scores. So people with good vision, which does also obviously limit the possibility of improvement somewhat if you're starting with good vision. Next slide, please. So this is the modified intention to treat analysis, showing that the photobiomodulation patients treated and planned to be treated and treated a bit and then interrupted overall were getting about 2 letters of improvement, but there were people that missed treatments. Next click. And this is this is just looking at the patients that received all the treatments they were planned to receive. So a lower number of patients, unfortunately, but there are 29 patients, but it was statistically significant. The difference between sham and photobiomodulation, the photobiomodulation patients being about 4 letters better versus about half a letter, it looks like, on the sham group. So there was a difference that was statistically significant in the visual acuity at the end of the period for those that we managed to get into treatment. Next slide, please. So this is, there were some patients that did particularly well, and again, this just shows that more patients as a percentage did better, getting more than 5 lines gain compared to those in the sham group. Again, I don't think this was statistically significant, but it's just interesting to see, and again, a sort of signal that maybe something's happening. Next slide, please. And this is the mask contrast sensitivity, which is another of the things we were looking at. It's slightly difficult to interpret this, the problem being that the photobiomodulation patients that completed treatment had quite good contrast sensitivity to start with, and the sham group slightly worse than the photobiomodulation group. So although the sham group did improve a little bit with time, the photobiomodulation didn't. But it didn't get any worse either. So it probably not really very meaningful either way, that with the low numbers. Next slide. So we'll be aware that with dry AMD, people often start off with drusen and thinning of the choroid capillaris and end up with atrophic patches and missing photoreceptors and retinal pigment epithelium. So one of the things I'm very interested in is whether we can reduce the amount of dry AMD. The other problem that we have with other treatments that have been tried for dry AMD, particularly from Apellis and in the Derby and Oak study, and also in the GATHER1 trial, is that they've all reported much increased rates of development of wet AMD. So although they seem to have some benefit, those injection treatments into the eye in reducing dry AMD, the wet AMD gets worse. So if we have the next slide. So one of the things we were looking at was the rate of conversion to wet AMD. What this shows is that the high risk for conversion, because a lot of these patients had wet AMD in their other eyes, particularly from our centre, you might expect some of them to develop over a year's time wet AMD in their PBM treated eye or sham eye. But in fact, none of the high risk patients having PBM developed wet AMD, which is kind of reassuring because I was certainly worried about that. Next slide. And in fact, the overall conversion, the numbers here are very small, so not statistically significant, but just again interesting that the photobiomodulation treated eyes had, if anything, slightly less conversion to wet AMD compared to the sham eye or sham treatment eye or the non-study eye if somebody had no wet or dry AMD in the other eye, or no late AMD in the other eye. Next slide. So this is also very interesting, I think, the central drusen outcome. So drusen deposits, we know that the bigger the deposits, the larger, the more confluent, the soft type, and the more extensive they are, the more chance you are to develop visual loss over time. It doesn't necessarily follow that reducing the volume and drusen load means that you won't get visual loss. I mean, there are collapsing PDs and stuff, but nevertheless, I think if you say to somebody you can have a choice of lots of drusen or less drusen, they'd probably choose less drusen. So it was interesting that the photobiomodulation group, the blue line there, showed no progressive increase in the drusen load, whereas the sham group did gradually increase as you would expect. So there's an interesting potential for disease modifying benefit. Again, over 9 months, you're unlikely to really know for sure. Next slide. There was, again, a signal, again, not statistically significant, unfortunately, with the small number of patients. So we had a rather limited number of patients that had geographic atrophy of the ones that were treatment, 8 sham and 8 photobiomodulation. But again, the suggestion from the data was that there was a decrease in geographic atrophy in the PBM treated group at 9 months. So, you know, if this really does pan out with further studies and further data, then this really would be a very interesting thing, in my opinion. Next slide, please. So the safety analysis, next slide, pretty safe. Just because the way things are reported, you'll see choroidal neovascularization. There's 1 case in the sham group, and then further down there's neovascular AMD. There's 1 case in the PBM group. That was not one of the high risk PBM. It was not the low risk PBM presumably. So no real difference there, but certainly no sort of danger signal coming up in the patients that we've seen there. Next slide, please. And then, yeah, there's a lot of kind of information there, but it's mostly things like hiatus hernia and carpal tunnel syndrome, stuff that we're really not linking in any way, but you just have to report them anyway. So we couldn't find, or I'm not aware of any significant danger signals. It seems to be a pretty safe thing to do, albeit we need more data. Next slide, please. So this is the sort of the take-home message slide, if you like. So sustained visual improvement, the primary endpoint at 9 months was statistically significant. 4-letter visual improvement at 9 months from baseline for patients that had all their treatments. So 3 lots of 9 is 27 photobiomodulation treatments and 30 eyes. So that was statistically significant. The other things that are really interesting, but we just didn't get enough patients to really demonstrate statistical significance, was the geographic atrophy, decreasing 44% below what would be expected. With the PBM and also the drusen growth, again, just seemed to stop the drusen growth progressing in photobiomodulation compared to sham, which, you know, if that's true and pans out to visual, to sight saving over time will be really interesting. We didn't find any change in the conversion from dry to the wet form, which has been shown in some of the other studies, and certainly something we need to look out for with the longer follow-up safety studies and registry studies that are being run in Europe, but overall no safety concerns from the data we've seen. I think that's me done. Unless anything else, Clark?
Yes.
Oh no, not quite. Sorry, just wanted to show you how easy it is to treat people. So yeah, so we're getting patients to come in, and once we got it up and running, we had them. They would basically arrive in their taxi. They would nip in, they would have the treatment for 5 minutes, the taxi meter would still be running pretty much, and they'd just nip out and get back in the taxi again. So they were really whizzed in and whizzed out. Next slide. And the next slide. So, yep, I think that's it then. Thank you very much. Great.
Thank you very much, Ben. I also want to point out to those on the webinar tonight that at the bottom of your page, there's a button called Q&A, and if you have a question, you can click that and add your question anytime. We will field those questions at the end of all the presentations, so you have an opportunity to think about it, or you can plug one in there and it'll just wait in the queue for the end of the webinar. Thank you, Ben, for your presentation, and I'll pass it on to Clark and get out of the way here.
Thank you, Dwayne. Ben, that was excellent summary of LightSight 2. And to continue on from that thought, unfortunately, obviously we had planned LightSight 2 and LightSight 3 to be kind of sister studies, one in Europe, one in the US. LightSight 2 was initiated prior to LightSight 3 and kind of ran right into the teeth of COVID. We did shut that study down for about 11 weeks while patients couldn't get into sites in some of the centers, and we had conferred with all the investigators to do that. When we brought it back up, we were concerned about fall reemergence of COVID, and so we decided just to let the patients come on through the study. And in the meantime, LightSight 3 was starting a little bit later in the US. There was probably more of a blend of academic centers in the UK than in the US. So as COVID came into LightSight 3, most of our centers were able to adapt and restrict patient access and minimize the risk for the patients. So we didn't have as huge of an impact because of timing in LightSight 3 than we did in LightSight 2. But I think both studies are very similar. In terms of inclusion criteria, they were very similar. They are both dry intermediate type studies. The centers here in the US are shown. LightSight 3 did enroll 100 subjects, 148 eyes qualified, so each eye could qualify individually. If they're randomized into either a PBM or a sham treatment group. The investigators here, you can see Stanford with Diana Doe and Quan, her husband, Richard Rosen, New York Eye and Ear, Eleanor Ladd at Duke, and I would say that the imaging centers work together in Europe. Marion Monk and the Bern Imaging Center was a central reader for all the sites. She coordinated then when we launched the US study with Glenn Jaffe at Duke, and Duke was the imaging center for the US trial. And I think both in Europe and in the US, we were very excited to have these centers all involved with us and continue to reach out to them as we move further in the research and development of this project. So the LightSight 3, similar to Europe, it's a double-masked, randomized, sham-controlled, parallel group multi-center study with 10 centers to look at both safety and efficacy of PBM in subjects with intermediate dry AMD. Similar to the European study, we had an active PBM group with the 3 wavelengths or a sham treatment group. And the sham treatment group is really an active control. We reduce the wavelengths 590, the fluence is reduced about 50-fold. 660, we reduce 100-fold. And the near infrared, since nobody can see it, we can turn it off in the sham. However, it is active, and as you saw in LightSight 2, and we've seen in LightSight 1, there is stimulation. The patients can see the activity at the photoreceptors. Those same cells, the photoacceptors, such as cytochrome C oxidase, are getting some signals. So it's not a pure placebo, but it's very difficult to mask light-based studies. And so we had fluences that were bright enough to allow the study to be masked, but hopefully not have an impact on disease progression. The study included BCVA from 2032 to 2100, and as Ben had said, no central involving GA in the central 1 millimeter fovea. The treatments were similar every 4 months as in the LightSight 2 study. We did extend this out to 24 months. And so for these patients, they came in every 4 months for 6 rounds of a series. After month 13, the patients had exited, the study was locked, and what we're going to present is the top-line data from the primary efficacy month 13 data. We did have a second primary efficacy at 21 months if the 2 groups did not separate, but we saw a nice significance at the month 13 data with a p-value of 0.02. So it allowed us to unmask the study, and we're continuing to look at the data as it's emerging. Primary endpoint was the 13-month comparison between the sham and PBM. The secondary endpoint was the intra change in BCVA within the PBM group. We also included, for the first time, low luminance, central drusen volume and thickness. We've seen reductions in multiple studies. That's involved as a secondary contrast sensitivity. And we also included some other exploratory endpoints that we'll be able to talk about today as well. The characteristics of the 100 subjects, 148 eyes, 2-to-1 randomized, PBM to sham, predominantly Caucasian, about 68% female. The mean age was around 75 years, similar to the European trial, but it was a little bit later. So the European trial was under 4 years, and here we're a little bit later, around 5 years from diagnosis. And in the US study, we have about 86% of the patients are taking ARID supplements. We did stratify versus ARID supplements, so both groups would receive an equal balance. And in the European trial, it was down around 30% that were on the ARID supplements. Similar to the last study, the baseline BCVA baseline letter score was pretty good, 70 letters in both groups, so around 20/40 starting vision in these patients. So top-line data showed that at month 13, we did see a statistically significant difference between the PBM group and the sham treatment group, p-value of 0.02. We also looked at from baseline to month 13, a very significant improvement in BCVA with about a 5.5 letter gain, p-value of 0.0001. And you can see it was a sustained benefit with the every 4-month interval, with significance at 1 month and pretty much throughout the study design. I will say at month 13, it was significant. We got about a 2 to 3 letter gain, and we've seen this in the past. We'll go through and look at, we do know that some patients can be very sensitive to low levels and that they do track as if they had a treatment benefit. So this is something that, because of the trial design, we do give away with an active control about 1 to 2 letters. When we look at the distribution across the 2 groups, we see in the PBM group a very nice shift to the right towards improvement in letter score and best corrected visual acuity. And you can see that from the green in the PBM versus the sham, and that also reflects a decrease in the amount of patients that have letter loss. To look at that a little closer, if we look at patients that got at least a 5-letter gain, we see about 55% of the patients get a 5-letter gain, and that mean for that group is about 9 letters, compared to about 40% of the sham. If we look at, I think even more impressive with the starting vision, is that we get about 1/4 of the patients or more that get at least 10 letters versus about 14% in the sham. So we're clearly seeing a significant number of patients that are getting a BCVA letter improvement. Now, conversely, if you look at letter loss, while the numbers are pretty small, we do see about twice as many patients in the sham that are having some letter loss versus letter gain, and twice as many that have letter loss versus letter loss of PBM. We had one patient that lost 20 letters. This was a patient that did have a cataract that was worsening, and we will do a stratification for those, pre-specified stratification with those patients that had worsening cataract. We hadn't looked at low-level BCVA, and we did look at it at month 1 after the first round of treatment and month 13. There was really no improvement in low-level BCVA, and in discussions with Nora Ladd, who's tracked this in earlier studies, it may not be the best tracking mechanism for early patients or with intermediate dry AMD. Overall, if you take a look at what we saw in the PBM as well as in the sham group and compare it to patients that were in one of the largest or 2 of the largest studies that followed GA disposition and growth, the Chrome and Spectra studies, you see that within a couple of years, you're going to be at where those patients were, and those patients lose about 5 letters over the course of 48 weeks. So really, I think is indicative that if we start earlier where these patients have viable tissue before we get to GA, either unilateral or bilateral, we have an opportunity to shift the vision to an improvement in letter score versus a loss as we get later on in the progression of the disease. Contrast sensitivity, similar to the last study, LightSight 2, was not significant. Both the Europe and European trials will use the Mars contrast sensitivity. Patients typically in 65 or over, if they're around 1.42, that's considered normal vision. So these patients had pretty good vision. Now, we did see in other studies using the FACS, which is a little bit more sensitive to early vision, we see improvements in contrast sensitivity, or with patients that have lower contrast sensitivity and electrolyte, we can see benefits. So we'll continue to evaluate this, but I think this is indicative of the early good starting vision in these patients' population. In terms of drusen and volume, we looked at over the course of time, similar to what we did in LightSight 2. We see over the course of time, typically an increase in drusen volume in the sham, where virtually no change in drusen volume was seen from the beginning to the end of the study in the PBM treated groups. Now, in both studies looking at intermediate patient populations, the amount of drusen is smaller versus later stage. And so it didn't turn out to be statistically significant at 13 months, but it was starting to trend in that direction. If you look at the absolute levels, whether it's in millimeter cubed or change from baseline, you can see from baseline to month 13, we are seeing an increase in drusen volume in the sham patients, where virtually no change in the drusen deposition in the patients with PBM. And this is consistent with the LightSight 2, so we may do some meta-analysis with this set of data. We are seeing a very subtle but in negative, a significant correlation, so that while the patients that get PBM do increase in visual acuity, they're really not showing a change in drusen deposition. In contrast, the shams are seeing an increase in drusen volume and limited improvement in vision. And so there is a correlation. It's not a one-to-one. You may not get an impact of drusen on BCVA, but clearly I think if we're making this tissue healthier, removing the hallmark, it can have a influence indirectly on vision improvement. This is geographic atrophy, and again, very small number of patients with non-central GA here. So there isn't much to talk about, except that you can see we start out very low in GA lesion area. We are seeing an increase over GA growth in the second half of the series, and does look like it's trending similar to what we saw with the LightSight 2, and we'll include these into a meta-analysis. But regardless, I think if we're making the tissue healthier, we would anticipate some disease-modifying effects on GA as well as on drusen deposition. Again, this is a GA showing change area change from baseline. The safety for this study, similar to LightSight 2, there are really no significant safety signals when we look at ocular adverse events. It was randomized 2-to-1 to PBM. We had about 3 to 4% conversion in neovascular. If you would compare it to a similar patient population, about 5% in the PBM. In the non-study eye, it's actually, again, was a little bit higher, around 6 to 7% conversion. And these are well within, I think, normal ranges for this stage of disease, below the 10 to 15% that we're seeing in some of the other studies. Other ocular, administrative, sight conditions, general disorders, infections, or infestations, really nothing. So the safety profile, again, in this study is out through 13 months and beyond. So some patients, about 30 or more patients, have already crossed and exited the 2-year study, and we continue to see a very good safety profile in all of our studies with the Valeda Light system. So in summary, we did meet the predetermined primary efficacy endpoint with a significant difference between PBM and sham at month 13. Very robust shift in terms of improvement in BCVA, with over 55% showing about a 5-letter gain with a mean of around 9 letters. 26% showed over a 10-letter gain with a mean of about 13 letters. And really, nothing in terms of contrast sensitivity or low-level BCVA. The patients had fairly good vision. We are seeing some consistent effects and reduction in central drusen deposition throughout the course of the study, and an excellent safety profile. So with that, I'm going to stop and allow some questions to be discussed from the Q&A.
Perfect. Thank you, Clark. Let's just go there. The first question is, how many patients have been treated in all the studies done on Valeda?
That's a great question. So we've treated over about 200 patients, depending on LightSight 1, 2, or 3. We had 2 studies prior to the Valeda that were part of the clinical evaluation report for the CE mark. So we've run now 7 studies, 6 have been in dry AMD patients. And in Europe, we've treated a little over 4,000 patients over the last 3 years, collected in certain centers 200 to 300 patients. So we have a significant database that we're continuing to grow for dry AMD.
Excellent. Thank you, Clark. The next question is to do with the minimum time gap required between the 2 treatment sessions. And I think this one we can probably go 2 ways, Clark, and should probably do both. One is between individual sessions, and then in between the complete cycle. I think they both merit discussion.
So at least within the sessions, we try to create a fairly flexible 9 sessions over 3 to 4 weeks. In our US trials, over 3 to 5. But typically we say you don't need to treat every day. 3 times a week has been indicated is ideal. If you miss one, come in the 4th week. The idea is we're stimulating the retinal tissue. That creates an upregulation in not only cytochrome C oxidase, but in the electron transport pathway. And PBM has been shown to upregulate probably over 100, 150 different proteins and enzymes. So we're kind of resetting the metabolic activity. And to do that, you get repeat treatments in various therapeutic areas, typically 3 to 4 weeks, and then you get sustained benefits. On the sustained benefits, it can vary between acute or chronic disease. In chronic disease like AMD, we did our first study at 6 months intervals. And in Europe, we typically say between 4 to 6 months, have the patients come in. If they're starting to remiss, schedule another round of treatment. We saw at 6 months with patients that were highly prone to AREDS category 4, over 50% late stage patients, that some of them, about 13 to 20%, were remissing by 6 months. And so hence we moved the studies into 4 months in both Europe and US, but we also did something else. We took out those late stage patients with 2100 or worse vision, because we saw that central GA correlated with response. If the tissue's gone, we really can't fix that. They didn't respond or do as well. So what we've subsequently seen in the electrolyte study is some of these early patients still have improved vision 6 months out. So I think it does depend on where you're at in the course of the disease, and that will be evolved, I think, just like any other drug therapy that comes on. You understand that depending on where the disease is, you may treat differently. Ben, I don't know, you want to add anything to what your thoughts are from your experiences in the trial?
Sorry, no. I was just looking down the next question. How can patients join the study? I think they can't join the study at the moment, can they?
So we do have our CE mark. So we have a registry study that is getting ready to be teed up. We have centers that, like Ben, that want to participate in the registry study, and it allows those patients to also, that were in the sham, to come over and get treatment. So I would reach out either to Ben or some of the other investigators that have devices that'll offer the treatment. Commercially can be located on our doctor locator on our website.
Yeah, and then the next, oh, sorry, go.
Yeah, this is probably for both of you. Is there a role in wet AMD for photobiomodulation?
Well, can I take this one? I mean, I think that's a really interesting question. And what we know about wet AMD is that people, we're quite good at treating it in the short term, but we know that in 5 to 7 years, people lose vision, and the mechanism they're losing vision from is geographic atrophy over scar tissue. So there may well be, obviously we don't know yet, We have to be a bit wary that the other treatments, the complement inhibitor type treatments that help with preventing geographic atrophy or limiting geographic atrophy, do seem to encourage wet AMD, and that there is this link between sort of hypothesized link between choroidal neovascular membranes under the retina, giving the retina more oxygen. That might be a good thing if it's controlled, and if it gets out of control, it causes scarring and so on. And so there might be a sort of a balance where you have more wet and less dry, or the opposite. So yeah, there might be a role, but it would be great if I would just persuade Luma theorists to do a study on wet AMD patients with photobiomodulation, see if it. But you'd need a sort of a 5 to 7 year study. So I'm not sure we're going to know the answer to that for quite a while. But certainly, I've treated people with wet and dry AMD privately.
Yeah, I would just add to that it's an excellent question. Strategically, when we started, we felt that the patients with dry really had limited options besides AREDS vitamins and lifestyle choices. I'm a pharmacologist by training. I think the anti-VEGF drugs are excellent drugs, very revolutionary for ophthalmology. But as been pointed out, the underlying disease continues to march forward. So we did put a wavelength in there that does reduce VEGF expression in retinal epithelial cells to 590 and can improve phagocytosis, with the concept that it might be used either as a, in addition to Eylea or Lucentis or the anti-VEGF, it may be used as another potential to, once you've controlled the neovascularization, haven't got completely the vision recovery that you wanted, maybe there would be some benefit there. Obviously, there's patients that they're resistant, and there are other medications coming forward that are going to address separately GA growth and that. The nice thing about this, it's a local treatment. It's not adding a 7th or 8th drug to an elderly patient. So I think there are opportunities. We have to do the clinical trials, as Ben pointed out. I don't think it's a standalone, but I think it could be used to maybe extend, or maybe to use after you've controlled wet. So there, I think, is a lot of research here that could be done, and we're excited about expanding this as in other areas beyond just AMD. So
it's probably just worth saying that if you've got a patient who doesn't like having injections and says, "Is there anything else?" I don't think this is not something else for that. I wouldn't. Yes, that you could offer that, as this isn't going to cure wet AMD.
No, I would agree wholeheartedly. Our focus has been on dry. We've had a few doctors treat a few patients, but there is not enough data to support that. And I think that the anti-VEGF drugs are revolutionary. They are very powerful. So that's the first line of treatment that needs to be approached for wet.
Thank you. A little shifting gears, what is the data on multifocal ERG or microperimetry in the eyes treated versus the sham? Do we have any data?
We did a small study that's being written up for publication with diopsis called the electrolyte study. And the idea was to look for other biomarkers. We use imaging to identify where the patient is at, but that is still static, and we'd like to have a quantitative, measure of functional vision. And the ERG study allowed us to do a baseline ERG and then measure each week before their round of 3 days of treatment. And what we saw is over the course of the 3 weeks, we got an increase in retinal ganglion output. It was a significant, about a 15% increase in multiluminance area under the curve, improvements in fixed luminance, improvements in negative photonic response as well. And so what we saw was about half the patients actually maxed out around 2 weeks. So it may be able to use that as a quantitative marker to optimize the treatment. And then we followed the patients, and these were the same intermediate as in these 2 studies. And out to 6 months, they still had about a 14-letter gain and improvement in contrast sensitivity. So we do feel that it could be used to also monitor when you need to retreat the patient. So I wholeheartedly believe that imaging and other biomarkers can be used around therapies to monitor and make the best approach at treating this disease.
What about microperimetry, Clark? Have you seen any data on that?
Yeah, in Europe, actually, there's several centers in our light site. One, we use microperimetry, and in patients, we've seen, I think, consistently improvements in fixation stability in some of those patients that have fixation stability issues. You can see improvements in visual field. I like microperimetry as another quantitative marker, and I think we didn't use in the bigger studies. Not every center is good at it, but I believe it's a good output marker as well.
Ben, do you have any experience with microperimetry and PBM?
I haven't used it. It's quite time intensive, and there's a bit of a learning curve getting used to it. So no, we haven't used it yet. We're looking at it for another study actually. But
Okay. Yeah, I would agree, Ben. That's one of the reasons it's not in our studies. And then two, the later stage patients can have some fixation stability issues, so it's complicated to get a reading on those patients, and can be very, lengthy for the patient. So with everything that we're doing, we've kind of shied away from that. But we do see centers in Europe and in Canada that have used microperimetry and shown benefits. So it's if you have it in your office and use it, it's something that you can see some benefits with.
All right, thank you. Question is, there was only one non-Caucasian in the study shown. Was there any reason for this?
No, there really wasn't. I mean, our centers across the United States were fairly geographically diverse, from rural centers and outside of DC to New York, Florida, North Carolina, Southern Texas, California, Stanford. We do know that the patient population typically tends to be more female, a little bit more Caucasian in dry AMD. Unfortunately, some of the maybe bigger university centers, they did get impacted by COVID more than some of the private ones. So the centers in New York and California didn't get as many as we would have liked going in because of COVID and the university. So that may have had some influence on the overall distribution of patient demographics.
Ben, what was your experience with LightSight 2?
Yeah, well, I mean, I just live in an area that's predominantly Caucasian. If you see somebody that's Afro-Caribbean, Asian, whatever, they're probably a doctor or a dentist working at the hospital. I don't know why, that's just the population that we serve. So I have about 2,500 patients treated for wet AMD, and I don't think a single one of them is, not Caucasian. So there just aren't, I couldn't possibly have recruited any non-Caucasians, even if I've particularly been trying to, which, so it's just, that's the population I serve. And if you did the same study in London, you'd probably have a different outcome, or Bradford or somewhere, but not in Great Yarmouth.
Okay, thank you.
I guess it's an interesting question whether the results would be different in a population with higher pigmented, melanin absorption in the retina in darker skinned people. I don't know.
It's interesting. We do have, besides our CE mark, we are approved in several countries in South America. We seem to see benefits there in the investigators that are using it. In terms of the absorption characteristics of the wavelengths, that is a question that's been asked before. Typically, melon will absorb, but it isn't the peak absorbance for either near infrared 590 or 660. So when you look at the absorption peaks and spectrums, those longer wavelengths are kind of free from some of those other contributing factors. Color of the pupil, again, we do a 30 millimeter beam, so we are, it's a bundle of light Obviously, during the open eye, it's going through the pupil, but even in the 660, we have the patient close their eyes and we can see it go through the skin on the inner eyelid and can get through the sclera. We do know that these wavelengths can penetrate, the skin into muscle and is used in skeletal muscles. So my comment is that there's probably some absorption, but not enough to make a big impact on the overall fluence getting to the retinal tissue.
Okay, thank you. Another question is around the 590 nanometer wavelength. What is the rationale for this wavelength, and what is that we claim that we can suppress VEGF secretion? Any preclinical or clinical data?
Yeah, it was work published by Dr. McDaniel in retinal epithelial cells looking at a variety of wavelengths and their effect on VEGF and a couple other outcome measures. There's been also a group in, I think, around Denver area that's published on some of the 590 in terms of its ability to work efficiently in ischemic conditions and also improve phagocytosis. So there's a smaller body. Most of the technology and research uses red because it's easy to see and it's clearly the most favorite for research. Near infrared is also well utilized, particularly in the CNS where we were doing work earlier in my career because near infrared has the deepest penetration. But there is body of literature for all three, including the 590. That was what we were using to target those different wavelengths.
All right, thank you. Another question, are non-medical personnel allowed to deliver the treatment? I think probably we can handle this in two parts as well, Clark. One is obviously personnel within an eye doctor's clinic, and also possibly are we maybe in other non-medical people having the device and treating patients?
Well, maybe Ben, do you want to talk about how you
well, yeah,
treatment
we certainly have non-medical personnel. So Emma Stimpson, who I think is actually on the call, the webinar, does pretty much all my treatments for me. So and runs a very efficient service and did the research trials for us as well. So yeah, no, you don't have to be medically qualified to deliver it. You might want to consider what your insurance status is if it turns out that there is some unexpected side effect down the line. So the insurance for any sort of novel treatment, you just might want to check that you're covered for that. But the other thing is to say that these patients are by definition at risk of wet AMD as well, and just so some sort of OCT scan every 4 months or so might not be a bad idea, or questionnaire about, have you noticed any distortion, or make sure they're using an Anderson grid. So yeah, so I don't think it really matters who gives the treatment, but I think there is a sort of an issue about these are potentially quite high risk patients for developing other conditions, which, admittedly an optometrist could easily pick up with an OCT scan.
Yeah, I think one of the things here, just to expand on that, this is not a laser. We use a LED that's further homogenized and diffused so that we've tested each wavelength for eye safety, and it meets through all the industry guidelines and testing. However, we only sell to doctors. We do anticipate that this is something that can be used in the office by a non-physician under the supervision of a physician. And it could be something that optometry might be able to treat depending on the country and or the local regulations. We felt that this could be an extension for the doctors so that we preload, the wavelengths, do all the testing so that it is fairly easy to learn to do in terms of training. And I think Ben alluded to that earlier, that, a technician can learn it pretty well. But, our clinical trials and all of our customers are MDs, and we don't anticipate selling this to an individual person at this point.
Thank you. Thank you, Clark. Thank you, Ben. Listen, I'm almost at the end of our time. So I want to thank you both for being part of our Peer Perspectives program for photobiomodulation. I also want to have a little bit of a plug. I will be at the RCO meeting. Unfortunately, we aren't having a stand there, but I will be there Tuesday, Wednesday, and so if anyone wants to meet and go over some of this information you're going to there, I will be there. And you can reach me at drmorrison@lumathera.com. That's drmorrison@lumathera.com. And if you have any other questions outside of that, you can reach Attila Kaplansi at the email listed below. I just, again, want to thank our presenters and thank each one of you for being on this evening. And I wish you well, and we hope to see you live at a conference near you at some point. Have a great evening.
Ben, thank you. It's been, as always, it's fun getting on and talking with you about our clinical trials, and enjoy RCO coming up next week.
Well, thank you. Thank you for having me.